Hello friends, welcome back to my channel BioFric. In today's lecture video, we will be starting with the non-cyclic photophosphorylation, and in addition to that, we'll also look the Q cycle, how and what all happens in the Q cycle. Okay, so now in non-cyclic photophosphorylation, which is a part of light reaction, we are aiming to form the reduced form of NADP plus, that is NADPH. At the same time, we'll also see how the ATPs are formed. These both the things will be later used in the dark reaction. Okay, so now let's get started. So here we have two molecules of water that are oxidized to O2, and 4 protons are released. And 4 electrons will be further passed on to PS2. Who does this? Who helps in this oxidation? It is oxygen evolving complex. This oxygen evolving complex helps in oxidation of water. It has certain proteins like PSBO, PSBQ, PSBP. Okay, PSBP. So you can remember OQ and OPQ like that. And besides that, it also has certain ions like chloride ions, calcium ions, bicarbonate ions, whose role is not really known. So from this, we move on further. That is, the electrons are already now with the PS2 system, and when this PS2 system absorb the red light, that is approximately 680 nanometer, and that's how it gets the name P680. The system gets excited, and excited P680 system passes the electrons to pheophytin. Now you might wonder, what is this pheophytin? Pheophytin is another component, but then it is similar to chlorophyll molecule. The chlorophyll has Mg in its center. Remember, I gave you the detailed structure of chlorophyll in the photosynthetic pigment lecture. So there I mentioned that there is magnesium ion in the center, but here in the case of pheophytin, the only difference is there is no magnesium. Instead, the valency is fulfilled by two H plus H atoms, hydrogen atoms there, and the pheophytin structure becomes stable. So that's how we have pheophytin there. Then we start with the plastoquinone. Okay, then the electrons, those 4 electrons, although it's not mentioned here, just understand that overall 4 electrons are passed on there. So these 4 electrons will be passed on to the quinone. Qa is the primary quinone there. Okay, primary quinone. Secondary quinone is Qb. Now, where these quinones are present, quinones can be closely or tightly associated with the PS system. To show you that association, just remember this is your whole PS system, PS2 system, sorry for that. PS2 system has D1 protein and D2 protein. Okay, so if you go to see, the water molecule is donating 4 electrons, and these 4 electrons are first accepted by the tyrosine molecule that is present in the D1 protein of the PS2. Okay, now this D1 protein accepts those 4 electrons, passes on to the pheophytin molecule. Pheophytin passes those electrons to QA, that is the primary quinone that is present. Now QA is a part of D2 protein. QA later passes on those electrons to QB, that is again the part of D1 protein. And that's how we enter the Q cycle. Okay, then we'll continue with the Q cycle. But before that, let's understand the further two components there. So there is something called as cytochrome B6, and there is also cytochrome F. Okay, the cytochromes have heme in their centers. Okay, heme A, B, and C. It can be anything. So cytochrome F has heme C, while cytochrome B6 has B there. Okay, so besides that, there is also something called as risky center. This risky center, the name comes from the name of the scientist who discovered this. So there is a risky center. So this whole stuff here is actually cytochrome B6, B6F. So just remember that. And the next component there is plastocyanin. Plastocyanin is the mobile soluble, water soluble component there, which is a protein again, and it has copper in its center, and it appears bluish in color, it seems. And also this plastocyanin is mobile one that will collect the electrons from cytochrome B6F, and it will pass on those electrons to PS1. So now let's begin with the Q cycle. What is happening in the Q cycle? Before starting with the Q cycle, I want you guys to know that there is a plastoquinone there. So this is a plastoquinone there. Then comes your semiquinone. Then comes your plastohydroquinone, or it can be also termed as plastoquinol. Okay, plastoquinol and plastoquinol. So just remember all these terms. Now we will start. So what does your plastoquinol has? Okay, this is your plastoquinol. Plastoquinol has two electrons, okay, and two protons, while semiquinol that is there, it will have just one electron, one H plus, while plastoquinone won't have anything here. Okay, this won't have anything. So now we begin with the Q cycle. Now the first half of the Q cycle starts with this. Okay, so now in this Q cycle, we start with the plastoquinol. Now plastoquinol, as I said here, it has two electrons and two protons. Now one electron will be passed on via the high potential chain. Okay, this whole chain that is there is high potential chain because it is facing the lumen side, and you know that there is also more H plus ions. By the way, this is also termed as to be the positive side, while this is the negative side. Hence this Bn and Bp that you see is nothing else but the heme groups of these cytochromes. Now heme group positive and negative is because of the side to which they are facing. Okay, now moving further, as I said, now plastoquinone quinol, which is there, it is giving one electron to the plastocyanin. It will follow this particular path. As it gives that one electron, it also passes one proton to the lumen, and it itself is getting converted to semiquinol. Okay, semiquinone. Isn't it possible? Like it is losing one electron, one proton, and it's getting converted to semiquinone. Now, the semiquinone that is there, it has one electron, one proton. It will pass that one electron and one proton to heme groups, positive and negative, the facing inner side, facing outer side towards the stroma, and it will form plastoquinone. Now this plastoquinone doesn't have any electron or proton. It will go back, okay, from where it had come, okay, and it will join. There is a pool of plastoquinones there. Okay, there is a whole lot of pool there. There are different plastoquinones, semiquinones, and plastoquinols there. So it will join that pool. Now what is happening is that you have your plastoquinone there, just plastoquinone. Now this plastoquinone which is there, it comes from here. Okay, another plastoquinone, some other plastoquinone enters this system. Now one electron that was passed from this semiquinone will go and attach to this, and it will take in one proton. Now what is happening is it is getting converted to this semiquinone. So in the end of the first half of the Q cycle, by the way, this Q cycle that I'm talking about, it is actually divided into the two halves. Okay, so that is divided into the two halves. So the first half that you have, you end up with semiquinone. Okay, since it is a cycle and we are starting with plastoquinol, okay, we started with a plastoquinol, we will end up with the plastoquinone. How? Let's start with the second half. Now in the second half, another plastoquinol will enter the cycle. Another plastoquinol will follow the same high potential chain. Okay, this is the high potential chain, and this path is the low potential chain. Okay, so first the plastoquinol electron will follow the high potential chain. Plastocyanin will get another electron. It will also pass another H plus ion. Now it is getting converted to semiquinol. which is obvious. Semiquinol has another electron. That electron will be passed on to the heme groups which are there, and from the heme groups this electron will be received by the semiquinol. Now this semiquinol which was there in the end of the first half, it is the same one that I'm talking about. Here I said that plastoquinone leaves and another plastoquinone takes in that whatever. Just remember this semiquinone was already there near this side. This side is the input side while this side is the output side. Output side is the one from where you can take the exit. These components can exit that system. Input, as in the semiquinone was already there, it will receive another electron and it will also take in another proton. Now semiquinone accepting one electron, accepting one proton, what it will result into? What it will result into? Here it was losing one electron, right, and one proton. Here it became semiquinone. Another semiquinone is now accepting. This will again result into the plastoquinol. Quinol, right? So we end up again with the plastoquinol. In this whole cycle, if you notice that there are protons which are getting pumped into the lumen. Overall, for 2 molecules of water, it is said that through this cytochrome B6F system, 8 protons will be pumped into the lumen, plus 4 protons are already there. Which 4 protons I'm referring to? This 4 protons. So during the Q cycle overall, you will get 8 protons here and 4 protons will be already there. Overall 12 protons are present here. Okay, overall 12 protons are present here. Now let's finish with this Z scheme first, and then I'll show you how ATP will be synthesized. So we reached here. Plastocyanin is transferring all those electrons to PS1. Now PS1 that is here, this PS1 will get excited by absorbing far red light that is of 700 nanometer. Once it absorbs the 700 nanometer, it will get excited and it will go give these electrons to the AO. AO is nothing else but it is a modified chlorophyll. Later it will be passed on to A1. A1 is basically another molecule called as phylloquinone. Okay, this is another quinone only. Now this phylloquinone will accept the electrons. Phylloquinone can also be termed as vitamin K1. This is important. Okay, phylloquinone can also be termed as vitamin K1. Then comes your FES series. Okay, I'll just clear this portion. Okay, so that you guys can understand what I'm talking about. So now we are here. We are near this FES. FES is nothing else but it is a cluster of protein. There is FESX, there is FESA, there is FESB. Okay, there will be series of transfer of electrons. Finally, the electrons will be transferred to ferredoxin, which is again the iron sulfur protein, and ferredoxin will pass on those electrons to ferredoxin NADP plus reductase. Ferredoxin NADP plus reductase, okay, is basically facing the stroma side. Okay, and later all this NADPH and all will be released in this stroma. So it is reducing your NADP plus to NADPH. So I hope this overall Z scheme is clear. There is nothing much except for that Q cycle part. Uh, and I hope you understood that. Now let's look at how the protons are pumped. And I know all this while you were looking at a bright screen and screen, and now I'm entering almost the dark theme there. So just get it adapted to this, and I'll start with that. So here, till here, I hope everyone understood. Okay. Here 12 protons were already formed. 8 protons were pumped in this cytochrome B6F, and for 2 molecules of water 8 protons were released because of oxidation by oxygen evolving complex. Overall 12 protons I have that is present in the lumen. Now in the lumen, obviously so many protons are forming gradient, the pH will be low, and all these electrons will pass through FOF not CFO CF not CFO-CF1 pump, and basically all those protons will, as they pump out of this protein, okay, proton motive force will be there. So ATP which is there, it will be formed. Okay, so how many ATPs will be formed? Approximately 12 protons are moving, right? So approximately 4 ATPs will be formed here, and that's the end of the light reaction. Besides this non-cyclic photophosphorylation, we also have cyclic phosphorylation. Okay, so hopefully I'll be able to show you the cyclic photophosphorylation in the next class. So yeah, stay connected for that. And that was it, guys. I hope you find all these things helpful, and see you in the next lecture. Bye bye.